I'm Satiani. In this chapter, we'll talk about pharmacodynamics. Pharmacodynamics describes the action of the drug on the body, or, to use a famous term, pharmacodynamics studies what the drug does to the body. It explores the effects of the drugs on the body, studies mechanisms of action, studies how a drug interacts with receptors, enzymes, other molecular targets to produce some therapeutic effect. Receptor is any biologic molecule to which a drug or toxin binds and can produce a biologic or physiologic response. There are 5 types of the receptors: ionic channel receptor, G-protein coupled receptor, intracellular receptor for the steroid hormones, receptor tyrosine kinase, and non-receptor tyrosine kinase, also known as receptors for the cytokines. Examples of the ionic channel receptors are nicotinic receptor, GABA receptor, glutamate receptor, and certain serotonin receptors. Nicotinic receptors are coupled with a sodium channel. After 2 molecules of the acetylcholine binds to the nicotinic receptor, central channel opens, sodium goes inside and potassium goes outside the cell. This leads to the action potential generation, depolarization, and contraction of the skeletal muscle. This is the way we move. Every time you breathe, muscles in your diaphragm depolarizes and contracts via the ionic channel receptors. GABA receptors also work by the same way. GABA is the major inhibitor neurotransmitter in the CNS. It's coupled with the chloride channels. Entering the chloride inside the cell leads to the hyperpolarization of the cell and inhibition of the neuron. Benzodiazepines, barbiturates, and alcohol work by the GABA receptor. They activate it and produce inhibitory effects on the neurons. Another example of the ionic channel receptors include metabotropic glutamate receptors and certain serotonin receptors. They have no clinical role and therefore I will not talk about them. G-protein, or previously known as GTP binding protein coupled receptors, are called serpentine receptors. Serpens means snake in Greek. A shape of receptor resembles snake because they have 7 transmembrane domains which cross the membrane 7 times. Examples are adrenergic receptors, cholinergic muscarinic but not nicotinic receptors because the nicotinic receptors are ionic channel receptors, certain serotonin receptors, some peptide hormones, odorants, and visual receptors in the rod and cone cells in the eye. G-proteins have types that you need to know: GS, GI, and GQ. GS receptor is coupled with the adenylyl cyclase. This enzyme converts ATP to the cycle KMP, a secondary messenger that promotes phosphorylation by activating protein kinase A. This leads to the phosphorylation of the substrates. In the cardiac myocyte, it increases calcium inside the cell and leads to the contraction. Examples of the GS-coupled receptors are beta-adrenergic receptor, dopaminergic 1 receptor, glucagon receptor, H2 for histamine, and V2 receptor for the vasopressin. On the other hand, GI will inhibit the adenylyl cyclase. Examples of the GI-coupled receptors are alpha-2, muscarinic type 2, and dopaminergic type 2 receptor. You can easily remember these: GS for stimulation, GI for inhibition. GQ protein activates phospholipase C. This leads to breakdown of the phosphatidylinositol bisphosphate into deacylglycerol and IP3. IP3 increases calcium release from the endoplasmic reticulum. Then, together DAG and released calcium will activate the protein kinase C, which leads to the smooth muscle contraction. Examples of the GQ-coupled receptors are muscarinic 1 and 3, alpha-1 adrenergic, angiotensin 2, H1 for histamine, and V1 vasopressin receptor. We'll talk about these receptors in detail in autonomic pharmacology. To avoid confusion, please remember that the cyclic GMP is not a secondary messenger for the G-protein receptors. Cyclic GMP is a secondary messenger for the nitric oxide pathway. Nitric oxide is a major vasodilator mediator which is synthesized from the endothelium of the blood vessel. From the endothelium, it diffuses to the smooth muscle of the blood vessel, enters inside the cell, activates guanylyl cyclase, which accumulates cyclic GMP. This will lead to the dephosphorylation of the myosin light chain and leads to the vasodilation. Intracellular receptors are particularly for the lipid-soluble drugs and hormones, such as glucocorticoids, mineralocorticoids, thyroid and gonadal hormones, and vitamin D. As you remember from our previous conversation, a drug must be lipid-soluble to cross the membrane. Steroid hormones are made from cholesterol. They are very lipid-soluble. They cross membranes and enter easily in every cell. That's why they have intracellular receptors. After entering the cell, hormone binds to the intracellular receptor. Then, together, hormone and receptor migrate in the nucleus. They bind to the DNA and change the gene expression. new messenger RNA is formed and new proteins are synthesized. This takes time. That's why pharmacological response on changing gene expression has slow onset, usually from 30 minutes to several hours, and usually hormones have long duration. Effects can persist from hours to even several days. Receptor tyrosine kinases are ligand-regulating transmembrane enzymes. Examples are insulin receptor, a natriuretic receptor peptide A. It's a receptor for the atrial natriuretic peptide, also known as ANP, and brain natriuretic peptide, also known as BNP. Growth factors like EGF, PDGF, and TGF-beta work by this way. In all these receptors, the two domains reside in the lipid bilayer of the plasma membrane. Binding the receptor can cause a conformational change. We call it dimerization, or a joining of the two receptors. After that, a cytoplasmic tyrosine kinase domain becomes phosphorylated on specific tyrosine residues and activated. This will catalyze a phosphorylation of the substrate proteins. We have anti-cancer drugs that work on these receptors: imatinib, gefitinib, erlotinib. They are tyrosine kinase inhibitors. They share a similar ending, TNIB, and that makes it easy to remember. A final type of the receptor is non-receptor tyrosine kinase. These are receptors for the cytokines, and it involves JAK-STAT pathway. Examples are growth hormone, erythropoietin and thrombopoietin, somatotropin, prolactin, and immunomodulators, including cytokines, interleukins, interferons, and granulocyte colony stimulating factors. JAK stands for Janus kinase. JAK contains two near-identical phosphate transferring domains. One domain exhibits kinase activity, another one negatively regulates the kinase activity of the first. The name comes from the ancient Roman mythology, a two-faced Janus. By the way, a January is named after it. Whereas STAT stands for signal transducer and activator of transcription. These receptors are on the plasma membrane as monomers. When cytokine binds to the monomers, they join and undergo dimerization. JAK kinase is activated and phosphorylates the STAT. Then a STAT-STAT dimer is formed, which enters in the nucleus and changes gene expression. I know what you're thinking right now. This receptor is like a combination of the insulin receptor and the steroid receptor. What I mean here is that JAK-STAT pathway is a combination of the receptor tyrosine kinase and a combination of the intracellular receptor, because at the end it migrates to the nucleus and can change the gene expression.